where are we going with this technology? We've got some fantastic data generated both in the basic research and also as it moves further forward. I guess one of the things that always strikes me going through this is there a point at which there is too high a plex? And I think we've got some data in there that suggests maybe there is and maybe there isn't. So I wonder if I could get some comment from the different people in here who have been testing that. Sergio?
I think if you provide me with a team of bioinformaticians, then my answer would be no. There is no limit to that. I think handling of data is a limitation. Yeah. So we need algorithms, proper ways of analyzing the data, because we are very good at generating data. I think we have shown this quite clearly today. Probably not my 7,440 genes, but your fantastic work with thousands of molecules and time points, etc. So to me, this is...
There's a team of bioinformaticians, as you must have. Okay. There's that.
It's a yes and no. Mainly no. You can't have too high a plex. I think for when you have a focused question or a specific application in mind, then certainly a few may be sufficient. But I think much of what we've seen so far is the power of these technologies for a discovery exercise. And then we do know what we're going to get out. A lot of it is going to correlate, but we don't necessarily know what the most informative features are going to be. And that's why we're doing the discovery exercise with a technology like this in the first place. So there will always be the demand for very high plex, I would imagine. And I think actually analysis is a challenge, but I think it's more wishful thinking that too high a plex is an analysis problem if we have enough good antibodies to look at everything that we want to look at. That would be my probably main limitation.
Any other comments? Dissensions? Any other comments?
Okay, so what the comment that I have is something I share with you in some other occasion. It's always in Roche, at least from what we can do, we always struggle to keep up with the technology. I think I met Dan for the first time 2 years ago, with Sarah, and since then, it already changed so much the technology that for us it's difficult also to predict whether there is a limit on multiplexing. But I think the real question is to ask whether we need or not the multiplexing, right? Because sometimes most of the question can be addressed with very few parameters, right? And whenever you scale up to the multiplexing, you need to have an enormous amount of work from the bioinformatician. And I think it's not that it's not enough. We were discussing yesterday about it. Also, you need to find a way to validate the parameters. I know that some people might not be agreeing with this, but I think there is an enormous amount of work after. And I think your presentation now was actually very nice to give a few examples where you really use a lot of parameters together to find a clinical output. And I think that is where a lot of work is still missing, I think, and it's very interesting for us to get there.
I guess the first thing I would like to say, it's always the question that you want to address that will drive the number of markers that you would like to measure. And in a moment, or just for exploration, it would be nice to have as many markers as possible, which is not possible given the limitations of antibodies, technologies, etc. But then there are also some nice studies, for example, out of Aviv Regev's lab, that show that once, for example, on a transcript level, once you have a comprehensive study, in the end, you only need about 200 transcripts to define all of the different cell states that are present in a sample. And so once you know this, you don't need this unlimited plex anymore, but you can really focus on what best describes the whole system.
Maybe I want to add a provocative comment, and that is that I'm not sure we already know what are the right questions. I think many of our questions that we ask today are based on a highly reductionist and limited view we have built up over the last 50 years. And so I think these data are going to give us insights into questions we are just currently not yet thinking of. But having said that, so I really think it goes really back to the fundamental basics of how we study biology. And indeed, then you cannot know a priori which markers to look at. Once you have done the exhaustive analysis, which unfortunately with proteins is not yet really possible, you of course can limit. That would be my comment.